天呐，哎呦喂， 哎。 呵。 是我记错了，好久没有。难道不是吧？Genomics. Isosomes. 是 immunology。哦。
From idea to submission, empower students to do their best work with Grammarly's comprehensive AI writing.
A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species. And so together with my colleagues, I've called for a global conversation about the technology that I co-invented so that we can consider all of the ethical and societal implications of a technology like this. What I want to do now is I want to tell you what the CRISPR technology is, what it can do, where we are today, and why I think we need to take a prudent path forward in the way that we employ this technology. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A big mouthful. You can see why we use the acronym CRISPR. It's a mechanism that allows cells to record over time the viruses that they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection. And as my colleague Blake Wiedenheft likes to say, the CRISPR locus is effectively a genetic vaccination card for the cells. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called RNA, which is orange in this picture, that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus associate the bind to protein called Cas9, which is white in the picture, and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, as you can see here, the blue molecule is DNA. This complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. It's a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognize that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break is introduced. It's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. So if we were able to program the CRISPR technology to make a break in DNA at the position at or near a mutation causing cystic fibrosis, for example, we could trigger cells to repair that mutation. Genome engineering is actually not new. It's been in development since the 1970s. We've had technologies for sequencing DNA, for copying DNA, and even for manipulating DNA. And these technologies were very promising, but the problem was that they were either inefficient or they were difficult enough to use that most scientists had not adopted them for use in their own laboratories or certainly for many clinical applications. So the opportunity to take a technology like CRISPR and utilize it has appeal because of its relative simplicity. We can think of older genome engineering technologies as similar to having to rewire your computer each time you want to run a new piece of software. Whereas the CRISPR technology is like software for the genome. We can program it easily using these little bits of RNA. So once a double-stranded break is made in DNA, we can induce repair and thereby potentially achieve astounding things like being able to correct mutations that cause sickle cell anemia or cause Huntington's disease. I actually think that the first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues. Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented litter mates by just a tiny change of one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology. Additional experiments are going on in other animals that are useful for creating models for human disease, such as monkeys. And here we find that we can use the system to test the application of this technology in particular tissues, for example, figuring out how to deliver the CRISPR tool into cells. We also want to understand better how to control the way the DNA is repaired after it's cut, and also to figure out how to control and limit any kind of off-target or unintended effects of using the technology. I think that we will see clinical application of this technology, certainly in adults, within the next 10 years. I think that it's likely that we will see clinical trials and possibly even approved therapies within that time, which is a very exciting thing to think about. And because of the excitement around this technology, there's a lot of interest in startup companies that have been founded to commercialize the CRISPR technology, and lots of venture capitalists that have been investing in these companies. But we have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties, such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Right now, the genetic information to understand what types of genes would give rise to these traits are mostly not known. But it's important to know that the CRISPR technology gives us a tool to make such changes once that knowledge becomes available. This raises a number of ethical questions that we have to carefully consider. And this is why I and my colleagues have called for a global pause in any clinical application of the CRISPR technology in human embryos to give us time to really consider all of the various implications of doing so. And actually, there's an important precedent for such a pause from the 1970s when scientists got together to call for a moratorium on the use of molecular cloning until the safety of that technology could be tested carefully and validated. So, genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.
Look, you're not actually going to get anything done by
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can end.
If you're a Unit 3-4 biology student and you've been trying to get your head around CRISPR, you may well be a little bit confused. And you can be forgiven for that because I'm sure, as you found, when you go out on the internet and you look at websites trying to answer those questions that you have about how CRISPR really works, you've found 2 kinds of websites. Some websites are presented really for the general public. They explain how CRISPR works in a general sort of way, but they don't really answer those nitty-gritty questions. They don't get into enough detail to help you really understand how it really works. And then there are other websites that clearly have been written for molecular biologists. And you read those and you're just met with a wall of terminology that makes them impenetrable. What I hope this video will do is bridge the gap between those two. We're going to go deep. We're going to look at the fine details of CRISPR-Cas9, how it happens naturally in a bacteria as part of their adaptive immune response, and how it can be used in a laboratory by geneticists to edit DNA. We'll look at all of that. And we'll go into more depth than probably you want to go into. But we'll do it slowly. We'll take our time and I'll explain all the terminology as we go along. So sit back and relax and let's get our head around this confusing topic of CRISPR and Cas9. Let's begin by talking about how CRISPR-Cas9 works naturally in a bacteria like Streptococcus pyogenes or E. coli or something like that. Essentially, CRISPR-Cas9 in a bacteria acts as an adaptive immune response. That is, it remembers when a virus has infected the cell in the past, and it keeps a little bit of viral DNA and uses it so that if the same species of virus infects the cell again, it will be able to respond to it much more quickly and more effectively. You can see here in my diagram that over here we have a bacteriophage. That's a kind of virus that infects bacteria, injecting its DNA into this bacterial cell like a syringe. It injects its DNA into the bacterial cell. And the first thing that happens is a pair of enzymes called Cas1 and Cas2. They are actually 2 separate enzymes, but they function together. They're joined at the hip, a bit like thing 1 and thing 2 in the Cat in the Hat. They're all laid together and they work in concert. And what they will do is they will cut out a region of the virus's DNA called a protospacer and stick it into part of the bacterial chromosome that's called a CRISPR array. Now, we'll talk about the CRISPR array in just a moment, but you'll notice that in the CRISPR array, there are repeats separated by spacers. And so this protospacer will become a spacer in the CRISPR array. The term proto means ahead of or before, and that's exactly what happens. You know, these enzymes, Cas1 and Cas2, identify this and think, well, that would be a suitable spacer. Let's turn it into a spacer. So it's a protospacer, and they make it into a spacer. Of course, they don't really think anything. They're enzymes, they don't have brains, but you get the point. Now, the spacer gets inserted at the 5' end of the CRISPR array. Right? That is, at the 5' end of the complementary strand of the CRISPR array. So it gets put there, and then they build a new repeat region afterwards. And you'll notice that there's a repeat after each spacer. Spacer, repeat, spacer, repeat, spacer, repeat. Every one of those repeats is exactly the same as all the other repeats. That's why it's called a repeat. And the spacers, of course, are in between them. The term CRISPR array, CRISPR, the word CRISPR, stands for clustered, regularly interspaced palindromic regions. And let's just break that term down a little bit. Clustered, of course, means all together because this CRISPR array is all in one place on the chromosome. Okay, all these spaces are all together in one cluster. Regularly interspaced is referring to the spaces that are regularly placed between these repeats along the CRISPR region. The palindromic repeats, well, the repeats part of that makes sense, right, because these repeats are repeated. And the palindromic bit simply means that there are regions within those repeats that read the same on both strands in a 5' to 3' direction. You might remember when you've learned about restriction endonucleases, that restriction endonucleases identify usually a restriction site because it's a palindromic. These repeats are often a site where enzymes can interact. For our understanding of CRISPR-Cas9 here, it doesn't really matter. It's not that important. That you understand where and how these things are considered to be palindromic, but that's what the word actually stands for. Now, when it comes to Cas1 and Cas2, taking a protospacer and turning it into a spacer, they don't just cut the DNA randomly in any old place. They cut it at a precise location just upstream of a protospacer adjacent motif. Okay. Now, a protospacer, this term, protospacer, is obviously referring to the protospacer. Adjacent means next to, and a motif that's an English word that just means a regularly repeated pattern, like you'd see on wallpaper or something like that. And of course, it's quite common in any DNA to find two guanines together, but the protospacer adjacent motif at least in Streptococcus pyogenes, is any nucleotide at all followed by guanine, guanine. Two guanines together, okay, following after anything, adenine, thymine, cytosine, another guanine, it doesn't really matter. Any nucleotide followed by two guanines, that is the protospacer adjacent motif, or the PAM, as people call it.
作为家长，你有许多必须知道，但学校不会告诉你的事，比如 VC 讲课时
Beautiful.
Okay, so what happens is these enzymes, Cas1 and Cas2, they'll scan the DNA looking for a PAM site, and when they find it, they'll go upstream, that is, to the 5' end on the complementary strand, or the coding strand, and then they'll cut out a section of bases, probably around about 20 to 26 bases long, and then turn that into a spacer. And as I said before, they'll insert the spacer at the 5' end of the CRISPR region and then build a new repeat to the 5' end of that, pushing the spacer further and further towards the 5' end. The CRISPR array then is flexible. As more, and if a particular bacteria has been infected by lots of different bacteria, it may have a very long CRISPR array with lots of spacers and repeats. Other bacteria might only have one or a few. I think in some bacteria, people have discovered that they have hundreds of spacers, and in other species of bacteria, there may only be a couple of them. Right? So it's a flexible CRISPR array. From time to time, RNA polymerase will transcribe that CRISPR region, that whole CRISPR array, into an RNA molecule. But we don't call it messenger RNA because it's not going to go to a ribosome and be translated into a polypeptide. We call it pre-CRISPR RNA. It's a single RNA molecule containing both repeat and spacer regions. And another kind of RNA called what's called unprocessed tracer RNA, which has been transcribed from another gene somewhere else in the cell. About a quarter of the way around the chromosome, there's another gene that's transcribed by RNA polymerase to make this unprocessed tracer RNA. And the tracer RNA has regions in it that are complementary to regions in the CRISPR RNA, and so it will stick to it. Those complementary regions will stick to the repeat regions in the CRISPR RNA, like I've drawn here. Then an enzyme called RNase comes along and cuts through those repeat regions, giving us a piece of RNA which is made of the RNA from the spacer, the RNA from the repeat, and some of the unprocessed tracer RNA, which we now call tracer RNA. So we have this structure in the cell made really of two pieces of RNA. One piece of RNA, one molecule of RNA, which is spacer RNA and repeat RNA. Okay, that's a single polymer of RNA nucleotides. And then we have another polymer of RNA nucleotides, which is held to it by hydrogen bonds, called tracer RNA. Altogether, we can refer to that as CRISPR tracer RNA, or CRISPR tracer RNA. Now that molecule gets picked up by an enzyme called Cas9. Cas9 grabs that CRISPR tracer RNA and holds onto it. And now we refer to it as gRNA, or guide RNA. Okay, so guide RNA is CRISPR tracer RNA, but it's CRISPR tracer RNA that's attached to a Cas9 enzyme. The Cas9 enzyme itself is made of a single polypeptide chain, but it has 6 different regions that do different things. In particular, we don't need to know what all these regions do, but there's a couple that I think are actually helpful to have a look at. You don't have to remember them, of course, but knowing them and appreciating them is, I think, pretty helpful. Firstly, there's this area here that I've coloured orange that is called the PI, which stands for PAM interacting domain. That's the part of the polypeptide, part of the protein, I should say, which identifies the PAM. Hence the name, PAM interacting. It identifies GG. It has a region in it that has a complementary shape and charges to 2 guanines, and that helps the Cas9 enzyme to identify GG something on a stretch of DNA. These two domains here are also really helpful. And by domain, we just mean a part of the protein. It sort of performs a specific function. It is all one polypeptide, though, again, just to reiterate that. But HNC and RUVC are what we call nuclease domains. That is, they're the part of the protein that actually cuts the DNA. A lot of the rest of this structure is there just to hold the guide RNA in place in the Cas9 enzyme. And this is what that looks like, or it's really a cartoon representation of what it looks like, because of course, the guide RNA is not all stiff like cardboard, it's all twisted and, and bent up. But it gives you a bit of an image of what it looks like there. You can see there we have both right here, the CRISPR RNA made of both the RNA that was transcribed from the spacer region of the DNA and the RNA that was transcribed from the repeat region, all as a single polymer of nucleotides, all as a single RNA molecule. And then held to that RNA molecule, we've got a separate polymer of nucleotides here, a separate RNA, held to it by complementary base pairing. The way to think about this really is the CRISPR RNA is kind of like the lenses in my glasses. It's the lenses that do the job of the glasses. It's the reason I have glasses in the first place is because of what the lenses do for me. The purpose of the arms of my glasses is just to hold the lenses to my face. And that's what the tracer RNA does in the guide RNA. The tracer RNA is really there just to anchor the CRISPR RNA to the Cas9 enzyme. It serves as a scaffold, we say, for the CRISPR RNA, holding it to the enzyme. You may have also heard of sgRNA, and there's a bit of confusion around that. A lot of people use the terms sgRNA and gRNA interchangeably, as though they're the same thing. They're not exactly the same thing. And there's even confusion around what SG actually stands for. Some people say that it stands for short guide. Other people say it stands for synthetic guide. And they both actually make sense because it is short and it is synthetic. And by synthetic, I mean it's made in a laboratory. But sgRNA stands for single guide RNA. Now, as I said, it's made in a laboratory. This is what geneticists do when they want to program Cas9 to cut DNA at a specific sequence, which we'll be looking at very soon, actually, is how we use this technology to cut DNA at a specific base sequence where we want the DNA to be cut. But when a geneticist is using Cas9 in this way, rather than using the natural CRISPR tracer RNA that happens naturally in a cell, it's easier just to make a single RNA molecule that will fold up and bend into the shape that they want it to bend into. That mimics CRISPR tracer RNA, but it's made as a single RNA molecule, hence the name single guide RNA. Okay, so if you hear someone saying that it's short guide RNA or synthetic guide RNA, they're wrong. It's single guide RNA. And it effectively, as I said, it does the same thing as guide RNA. In fact, it is a guide RNA. So we could say that single guide RNA is a guide RNA. Not all guide RNA, though, are single guide RNA. When Cas9 finds some viral DNA, or any DNA for that matter, and it identifies a PAM region, what it will do is it will open the DNA and then see whether the guide RNA that it has is complementary to the sequence of bases upstream of the PAM, but on the opposite strand. Okay, so here's our PAM right here on this strand. It opens the DNA and tries to match its guide RNA to the sequence of nucleotide bases on the other strand, but upstream of the PAM. And if it finds that the sequence is complementary, then Cas9 will cut the DNA on both strands 4 bases upstream of the PAM. Maybe 3 bases or 5 bases, around about 4 bases upstream of the PAM on both strands. Then, of course, that cleaves the DNA, causing what we call a double-strand break. A double-strand break, or a DSB, is how you'll see that written in the literature. After that's done, of course, the Cas9 enzyme then leaves the DNA and travels off looking for another PAM site and does the same thing over and over again, constantly looking for PAM site, opening the DNA, and then seeing whether the guide RNA is complementary to the sequence of bases upstream of the PAM on the opposite strand. Remember that a bacteria uses the CRISPR-Cas9 system as an adaptive immune response. Just like in our immune system, it's really important that our immune system is able to tell the difference between self and non-self. That's just as important for a bacteria. So how does that happen? How can the Cas9 enzyme tell the difference between virus DNA and bacterial DNA? Or to put it another way, if this particular Cas9 enzyme will cut this sequence of nucleotide bases on viral DNA, why won't it cut this sequence of bases on in its own CRISPR spacer region? Right? Because that sequence of bases is identical to that sequence of bases. And they're both DNA, they're both made of the same stuff. So how is Cas9 able to tell the difference? Well, of course, that comes down to the PAM site. Remember, Cas9 won't even interact with the DNA. It won't even open the DNA to see if its guide RNA matches the DNA unless it first finds a PAM site downstream of that region, right? And is looking for that PAM site. When it comes to the spacer region here, it won't even have a look to see if the spacer region is complementary to its guide RNA because this sequence of bases is not a PAM. It's a different sequence of bases. This is why CRISPR is so ingenious, is because each of these repeats has a sequence that starts with something other than any nucleotide followed by guanine. And that prevents Cas9 from cutting the spacer itself in the CRISPR array. Let's look next at how scientists are able to use CRISPR-Cas9 in order to precisely edit the genome of a species. There are two approaches to doing this. We call them non-homologous end joining and homology-directed repair. We'll look at both of those. Remember that time when your parents were out and you stayed home and you played footy in the house and you broke your mum's favourite vase, so you got some super glue and you glued it back together again, hoping that nobody would notice, but you knew that it wasn't really a perfect join. Well, that's pretty much what non-homologous end joining is all about. The non-homologous bit of the name will make more sense once we look at the next way that DNA can be repaired in a cell. But basically it means that you don't need other bits of DNA to act as a template. The end joining, though, is pretty obvious. It just means that the two broken bits of DNA can be joined back together end to end. Now, like most things in biotechnology, all the tools needed to do this come naturally from cells. It's the same in bacterial cells and eukaryotic cells. All cells are able to repair breaks in DNA because it happens actually surprisingly commonly. In any typical cell, there will be somewhere between 10 and 50 double-strand breaks in the DNA per day. That's a lot of breaks, and so cells need to be able to fix those breaks, and one way they do that is by non-homologous end joining. Let's have a look at how this works. Now, you don't really need to remember all of the fine details here, I wouldn't imagine, but understanding them and appreciating them, seeing how it works, will give you that depth of understanding to really appreciate how CRISPR is done. All right, so you can see here we've got some DNA that has been broken. It might have been just broken as a double-stranded break, an accidental break, or it may have been cut by a Cas9 enzyme. Either way, what will happen is a protein that exists naturally in the cell called Q7080 is very strongly attracted to the ends of a piece of DNA. And so Q7080 will stick on those ends, and it, in turn, co-opts attract another protein called DNA protein kinase catalytic subunit, which sticks onto the Q7080. And together, they attract, in turn, a bunch of proteins whose, again, names are weird, but XRCC4 and XLF. And those proteins kind of form, and this is obviously a cartoon version of it, but they form sort of almost like a spring around the two ends of the DNA and join them together. Sort of, you can imagine that happening. They sort of encircle them and bring the two ends together. And then another enzyme called DNA ligase 4 will join those two ends of the DNA. It makes a sugar phosphate bond between the sugar of one nucleotide and the phosphate of another nucleotide, joining the two broken ends back together again. And that sounds really great. And you might be sitting there thinking, well, why would a scientist want to do that? Because it seems kind of pointless. You use Cas9 to cut the DNA, and then the cell is just going to fix it back up again. And that's true. But just like when you broke your mum's vase, the superglue didn't join it back perfectly. And that's what happened here too. Sure, it gets joined back together, but often mistakes are made. Sometimes it's not perfect. Sometimes a few nucleotides are lost. Sometimes an extra nucleotide gets accidentally incorporated into the join. Of course, ligase, it has no knowledge of what it's doing. It's just a protein. It's just making sugar phosphate bonds between bits of DNA. And the process is a little bit sloppy or error prone, we might say. It often makes mistakes. As a result of that, a scientist is able to use this technology to hopefully induce a mutation in DNA that might knock out a gene. Now, you've probably heard of knockout genes and wondered what they were. Again, this is a point where there's a lot of confusion about this because people sort of assume that a knockout, when scientists knock out a gene, that what that means is they're sort of knocking it out of the cell. You know, sort of like knocking the ball out of the park in baseball, right? But it's not that kind of knockout. It's more like a knockout in boxing where the boxer who's been knocked out is still there in the ring, but they're just unconscious on the floor, right? That's what a knockout gene is like. It's a gene that's been kind of prevented from working. It's a gene that's become non-functional. And there's a couple of ways, of course, that could happen. But one way that I'm sure you're able to understand is just imagine that there was a sequence that went UGC, right? UGC. And you were able to cause a break after the G. Now, it's possible that an extra nucleotide might be added there. So, and if that was an adenine, it would now read UGA, which is a stop codon. And that would make that a knockout gene, or that gene would have been knocked out because we've just created a stop codon that would prevent it from then being transcribed and translated into a protein. We've knocked that gene out. Or a mutation in the promoter region of a gene would probably render it a knocked out gene as well. So there are a number of different ways that this could be used, but it's all based on the idea that while the cell will repair the DNA, it makes a lot of mistakes, and if it makes the right kind of mistake, that lucky accident can sometimes result in disabling the gene or changing the gene in a way that a scientist wants to do it. Another way that cells are able to repair a break in DNA is homology-directed repair. Compared to non-homologous end joining, this is a much less error-prone approach. So it's ideal for a cell to do this, but cells can't always do homology-directed repair. In order to do it, they need to have a homologous piece of DNA. That is a DNA with the exact same base sequence as the broken piece of DNA. Of course, in our cells, that's usually possible because we've got two of each chromosome. We've got a homologous pair of chromosomes. We've always got a backup copy of every length of DNA. In bacteria, it's often not the case because a bacteria's only got one circular chromosome, but it is replicated in preparation for binary fission. So at some times of the prokaryotic life cycle, prokaryotic cell cycle, there are two copies of each gene, and at that time, homology-directed repair can happen in a bacteria as well. There are two processes that can happen in a cell, both under the banner of homology-directed repair. The first of them is called synthesis-dependent strand annealing. It's the simpler of the two. They're both kind of variations on the same thing. We'll look at both of them, but let's have a look at this one first. You can see here, just while we're looking at it, that we've got a broken strand of DNA and we've got another piece of DNA that's homologous to it. It has the same sequence of bases on it. I've drawn it upside down. Now, the first thing that happens in homology-directed repair is what we call resection to achieve.
你找啦？怎么了？
要睡吗？
差不多吧，我现在在学呢。
明天干嘛？
明天也得学啊。
明天不是 CS 吗？
那也得要晚上才能玩。
他妈的那个真的真的不行，不好，太鸡巴那个难搞了那玩意儿。他让我验证那个东西，然后我他妈的验证好几次，他说我 ID 和那个我脸不符。
脸不符什么意思？
就他不是要那个验证那个账号吗才能玩？
哦，然后呢，你还要给 ID 是吧？
我每次就是我的驾照和我的脸，他说他不符，不过关。咋办呢？然后我跟驾驶员说，驾驶员他妈连下都没下，他说玩官服。
玩鸡巴官服，他妈，他搞不明白。
我现在他妈验证不了，我他玩不了，就这的。然后你他妈，然后驾驶员都不知道要交纳时看，你知道吗？他说交鸡毛。
他不懂啊，他不懂啊，载物动全都打飞起了，谁他妈打官皮啊？
他妈我们又不是载物，我们是动可吗？
不是，意思是说你在官皮上打了人都很傻逼的、啊，而且很多挂的，到时候他妈你没什么意思啊。
那我他妈我这通过不了怎么办？我操！
那真通过不了，那就试试吧，和官皮试试吧。
我操，那我这20块不白交了？
也不会，你那个绿色账号的话，你到时候你什么 CS 会给你枪，给你什么，给你箱子，然后呢，然后那钥匙什么的。
我这账号上啥都没有啊。
你要打游戏才会送，要干一小会，对，就小干一会，但是就像是那个什么瓦里的通行证一样。
哦。
对，然后而且是一开就是永远都开。而且你知道这个游戏夸张的是，它有点带赌博性质的，就是你在 CS 里开出来的那种枪皮啊，如果你开的运气好，什么绝世枪皮，可能他妈上千上百上千美刀都有，然后你可以交易，可以卖出去。
我知道那个圣狗的绿宝石不卖了10几万吗？
对啊，所以说呢，万一他妈的送你个箱，你一开，我操，卖阿密，或者是我操龙驹，然后你
这有可能。
真有可能啊，这不是开玩笑啊，不是说什么，就是它就是随机的箱子啊，它不是像腾讯那种死亡玩意，就是还要你要抽多少次不一样，它那个是你运气好就真的也爆了。
所以绿宝石真的那么贵吗？
真的呀，真的呀，还有什么
卖10几万人民币，
还有蝴蝶刀，还有那个什么对。
2-3万澳币。
你要不这样的话，你现在才知道为什么那够学长他妈去到网吧不敢登自己的号了，那库存里面都是他的命啊，跟股票似的，你知道吗？
去网吧登会被盗吗？
对啊，被盗号，然后直接把他清仓大甩卖了，是血本无归啊。对啊，够学长那个号里面那个库存哦，真的有些很颠的，很颠的，不是开玩笑。
我操！所以他们真的都是自己买的吗？
抽的，还有买的要是你还真喜欢，你要真喜欢。
他妈你一个刀他妈卖10几万人民币，他妈的这是
不是，你要听我说，是会涨到那样子，但是你要真的要找到买家，还得等一会儿，知道吗？就是你要看什么，如果你是出的那种很热门的那种，就是有价也有市，也有一些就是有一些是什么，就是你跟股票一样的，你知道吗？就是什么，就是它涨到一个点会跌的，为什么呢？因为其实是需求没那么大，你知道吗？就你涨到那么贵，那没人要买，你知道吗？也有可能这种情况出现，但是理论上价格就在那里，哦。嗯，
明天健不健身？
明天了都，明天的话怎么说啊？
问你啊，你要健你就他妈的喜欢做就过来健啊，我都行啊，我可以等你啊。
不是你要听我说一下，你要听我说，主笔，我想了想，我周3要演讲，所以说我周1练的时候不能练腿，我怕他妈的直接在场上都站不动的。
不会啊，腿都是最晚练的，就是一周的最后一天。
所以我先练胸没问题吧，然后我到时候周3的时候我不会说人废掉
啊，不可能的，你们想多了。
对啊，那没问题吧？
只要你每次你练完然后补充快碳都不会那个，这个精神状态不会很差的。
OK， 那给你看，注意我今天炫了一顿，我操，香了一批了。你看我自己整了一顿番茄酱意大利面配洋葱炒牛肉，我操，真他妈香啊！这不开玩笑，我他妈把我吃爽了这一餐。
那肯定香啊！我操！
你看这个肉是不是多了一逼？有没有问题？我这个量是不是没问题了？
一般
而且我放主食了。
如果是那个你每次健完身吃是完全没问题。
我已经放，我今天3餐就吃一顿面条，没问题吧？
没问题啊。
对啊，3餐里面2餐吃他妈肉和菜，一餐吃点面条，没问题吧？我这理解对了吧？我这理解不会再错吧？
面条不能放晚上吃啊。
我6点钟吃的没问题吧？
最好中午吃。
也可以，那行，那明天中午自己再下顿面，吧。
然后
我这个面条看起来就健康了。
那你就回家就不要吃了。
不是健完身要补快碳吗？屌你！
你就喝蛋白粉就行。
不行吧，大哥，练完不是要补快碳的吗？这不是公式吗？
你他妈要是你晚上8、9点去练，你再补充快碳，纯鸡巴长脂肪。
行，我估计明天没等，我明天也不一定。其实我现在把 PPT 做完，明天练可以，然后周一我再开会，可以的，也可以。我明天你几点的话，你想？
我早上去完教会我都行了呀。
你早上几点去完教会
？12点吧。
你的那个健身房的地址，还有没有停车场那边？有免费
停车场。
有免费停车不？
就免费的，他平时健身房自带的呀。
哦，行，那我赶紧写，然后明天那啥搞，我就过来。哎，不是啊，那我们就下午练呗，晚上就可以，我晚上还可以吃顿饭，不是吗？
这是最好的呀。
我下午练完之后，你说
只要我晚上练，我就喝蛋白粉，我不可能吃碳水。
那下午呢？
下午可以吃，你下午4、5点练完是没毛病。
练完我去吃什么？你说什么快餐比较好？
我说实话，你吃个 cheeseburger 完全没问题。
然后呢？一个 cheeseburger 就够了，是这个意思吗？
然后就喝蛋白粉啊。
我操，我没买蛋白粉，现在。
你明天去买不就行了吗？我操！这东西又不是说什么他妈绿宝石，我操，还要他妈的。
我不能回家自己，我主要是我冰箱里有些肉有些菜我不能不吃啊。
你可以回家做呀。
我回家做对啊，回家我就练完开车回家直接开做一顿饭，然后直接吃。
但还是要喝蛋白粉啊。
我狂吃肉还要吃蛋白粉。
你能他妈吃1公斤肉？
行吧，操，被你说服了。
啊，你这东西又不贵，我操。可能70、80能够吃半年。
行。
你在这上省是没道理的，好吧？而且你这种体格，你都不只要喝蛋白粉，你还要整别的东西。
我要整什么？加肌酸啊。
肌酸，然后氮泵也可以搞。氮泵就是练前喝，你更有力一点，因为你没肌肉啊，你没力啊。
氮泵是什么东西啊？氮
泵，你小红书搜就知道了。
操！ 他妈，我学到一半给你扣，那我操，我要不直接睡了，明天早上早起开始写算了。
可以啊，你不他妈每天都挺早起的。
对啊，弹蹦增强力量，what the fuck？ 这东西不是智商税，不是智商税吧？
是鸡毛，弄了如何？
行吧。
最适合这种，就是之前没练然后就喝，然后这种细狗。
细狗喝的。
对啊，就像宗人那种，就是骨架小的呀，你骨架也是小的呀。
我也算是小骨架嘛，对吧？
你他妈是你的肩和我的肩，你比一下就知道了呀。
哦，行吧。
但泵和那个肌酸就明天就也不用，就是你买完然后就是不着急，那个不是那么着急，但你蛋白粉是一定要练后就要喝的。
OK。
不然你吃不到，就等于说你花
那
的
冲多少喝，我这个得要自己想是吧？
不是啊，它有说明书的，每个人都一样的。
OK， 那
它
就是
后面有那个意思。
那就这样吧，我操，行吧，那我先挂了，我直接我下播睡觉了。
你明天啥时候来之前跟我说。
OK， 你把地址发过来吧。
OK。你先来我家呀。
我干嘛先来你家？